Good morning. How are you? I'm very happy to be with you this morning. So, my name is Hermann Prodjinoto. I was born in the most beautiful country in Africa, Benin, the place to be. Here is some monuments, a place is Benin. I did my PhD in Belgium. Now, I'm postdoctoral researcher in plant science at the Hebrew University of Jerusalem. I love superhero movie, but my own superhero is my mom. Please raise your hand if you also have a parent who is a superhero. Put all them together, and you will be close to the power of my mom. Indeed, when I was 5 years old, my parents divorced. My mother's education level was limited to primary school. Despite this, she succeeded in coaching me to become a scientist. Thank you, Mom. I remember when I was 8 years old, my mother and I traveled to visit my uncle in village. And I saw my uncle who was sad because his plants were thirsty. And there was nothing he could do about it. Plants were thirsty because there had been a drought in that village for several weeks. In the following days, the food prices doubled or tripled all over the country. That was a difficult time for my family. At that time, I learned a lesson. When plants are thirsty, people are hungry. Unfortunately, the drought problem is persisting until today. For example, when we look at the top 10 crises which the world cannot ignore, the first two places are occupied by the drought problem in Somalia and Ethiopia. In that country, people lost their lives to starvation, and these countries are on the brink of famine. That means that the lesson which I learned involved. Thirsty plants not bring only hungry people, but also dead people. Okay? But please, don't think that this problem is only for Africa. We all know that our population is increasing. That means that if we need to survive, you have to produce more food for ourselves to feed. Unfortunately, the fresh water availability will decrease by 50% due to global warming. So there is a question: how to grow, how to feed a growing population in increasingly dry world? The answer is you have to grow the plants in that increasing dry world. How? By improving plant water use efficiency. What does water use efficiency mean? Imagine after that conference, you decide to make some tomato salad or soup with your family. You go to market, and you found out that 1 kilogram of tomato costs $500. Will you be willing to buy it? I think no. Yet, this is what we have been doing in our agricultural system. To give you an overview, we spend about 500 liters of water to obtain 1 kilogram of tomato. That's not fair. Why? Because plants use only 5% of water it is given. That means 95% of that water is lost. This graph shows the correlation between the water supply and crop yield. In x-axis have rain and irrigation, and y-axis is crop yield. When there is enough water, you have enough food. But when drought occurs, the yield is not good. So if you want to improve plant water efficiency, you have to change that line. I mean, you have to have that thing little water to have enough crop. If you want to plant to grow in dry condition, you have to add an element through which water enters the plant. This is a cell membrane. In blue color is the channel through which water enters the plant. That channel are named aquaporin. Aqua for water, pour for pouring. So aquaporin. Aquaporin is the channel through which water enters in the cell. They were discovered by Peter Agri, Nobel Prize 2003. Okay? As far as tomato is concerned, scientists found out that tomato have 47 aquaporin genes. But there is a question. Is tomato really need all that aquaporin to have water to yield? Maybe tomato need 10 or 1 aquaporin, which will bring water to get enough fruit. That is my job. My job is to determine the role of each that aquaporin in water use efficiency of tomato to help tomato to become more resistant to drought. How I'm going to do that? I'm going to use a gene editing tool named CRISPR-Cas9 to delete one by one first aquaporin gene. This picture shows how I'm going to do it to have wild and transgenic plants. Okay? But I will apply different treatment of drought on that wild and transgenic plant. I mean, drought on wild fruit of life of plants, drought on the vegetative stage, in reproductive stage, and fruiting stage. But I have to choose on that all plants which plants are really relevant to resist to drought. That means that I need a system who can help me to make a good decision. That system have to continuously measure multi-key physiological traits such as transmission rate, root activity, and more. The same system also have to simultaneously control many different scenarios of drought. And third, this system also must help me to have comparative results in few weeks. Fortunately, I have this system in my lab. So this is how I will apply drought on my plants. I will collect all that data, as I said, biomass, transpiration, root activity. So I will have big data. I will analyze them, and then I can select which plant who will be very useful to grow in drought condition. I will do it in greenhouses, and when I select the best plant, I will transfer that plant in field condition to find out what will be the influence and the impact of the nature in that plant. I started this study a month ago. With some results I already have. I did some meta-analysis to find out what are the expression. The star color is the expression of each aquaporin gene in tomato. They are different in root, leaves, flower, fruit. I did some classification, and I found out that we can divide that in 5 different families, all aquaporin gene. Okay? And when we focus on the expression, when you compare the expression in roots and leaves, we found that there is different expression between roots and leaves. What means that the aquaporin gene expression in is organ dependent. But when we took one aquaporin gene and check what's happened in all tissue organ, what we have in some tissue is high expressed, other tissue in down expressed. That means that the aquaporin gene is also tissue dependent. So, ladies and gentlemen, what can we keep in mind? When plants are thirsty, people are hungry, some people dead or leave their country or village. If we need to solve this problem, we must improve water use efficiency of plants. How? By acting on aquaporin. So this will help us to boost plant drought resistance. That's what I wanted to share with you this morning. If you have any question or suggestion, please feel free to say it. I'll be happy to answer you.
What can you change? Can you? Sorry, I'm Jan from Tel Aviv University. Thank you for the talk, great talk. And which handle do you have on the Aquaporin? Can you change their numbers, their efficiency?
I don't get you. Can you repeat the question, please?
How can you? Which handle do you have on the Aquaporin? How can you impact the Aquaporin of a plant?
Okay.
Can you add Aquaporin? Can you improve their efficiency? What can you do as a scientist?
Okay. Aquaporins are naturally present. Thank you for your question. Aquaporins are naturally present on the cell. Okay. Currently, I just want to knock out them to check how their current role, the physical role. No one knows. You all know that they are there, but what are their role in plant? No one knows. We know that they help them to have water, but if all can bring water to plant, you don't know. For example, if I give you this aquaporin gene, in this gene, this is one aquaporin. This aquaporin expressed only in root, not in leaves. Overexpressed.
So you can modify the genetic of a plant to add aquaporin?
Yeah, I can add, I can delete, I can put another gene, I can improve the expression. So currently, I just want to do some, to check all their role by analyze the physiological role, the genetical role, the phenotypic role. That's what I'm going to do now.
Thank you.
Did I answer your question?
Yes, thank you.
Okay, welcome.
I have my name is Flavio from Hebrew University of Jerusalem. I was wondering about why tomatoes. Is there a lot of research on this, or do they grow fast, or what are some of the reasons? I mean, there must be practical reasons. Please.
Thank you very much. Tomato is the second most vegetable in the world after potatoes, and economically the fourth most interesting fruit who influence the world. So economically and healthily is very useful for all people, and we can find tomato in all continents. Europe, Asia, Africa. So, and I think every day there's no dishes we cannot use without using tomato. So improving tomato is the way to help people to get easily food, because my aim is to make a place for our future generation. If we don't have tomato, how can we have, for example, pizza or our hamburger? So we need tomato for our dishes, for our salad, for our soup. Welcome.
Hi, Sarah from the Ben-Gurion University. Thank you for the talk, very, very interesting. I relate with the previous question because I wanted to ask, so your findings on tomato then are generalizable to other species? And this is the first question. And the second is, how easy or difficult will be, in your opinion, to apply the changes that you're studying to actual agriculture, to the people that produces these kind of plants?
Can you repeat your first question, please?
Yes, if your findings on aquaporin is specific for tomatoes or can be generalized to other kind of...
Okay, thank you. Aquaporin is present on all cells. For example, as human, we have only 13 aquaporin in our cell. Tomato have 47. Other species have different number. We know that they are present. They help cell to get water. Not all we know. But what are their real functional and physiological role? No one knows that. Okay? So yes, we can apply that same system in other species, potato, and so on. So about farmers, yes, if I know by my study what are the role of aquaporin in the water process, in water supply in plants, I can use that knowledge to improve the water use efficiency of plants. And then I can select the plant who can easily grow in dry condition And in future, farmer can use that species, that variety, to get more food. So basically, it's some deeply research to really check what is the role of aquaporin. Welcome. Yeah, there's a question? Yeah.
People ask the question I have, so I have one. Thank you. I'm Rolando from Hebrew University. First, tomatoes already cost $500 in Tel Aviv. No, but no, it's more than a question. I want to understand. Aquaporins drives water inside the cell, but they also are used to release water. Because you were saying, I have 47 aquaporin. I want to reduce the number of active aquaporin. Why you reduce the number of aquaporin if you want to increase the efficiency? I didn't understand this part. I would expect you wanting more aquaporins to get more water if the efficiency is low.
Thank you, Ronaldo, for your question. All cells have aquaporin. And we know that some aquaporin are very expressed in roots, over in leaves, over in flower, okay? But we also know that currently we use too much water to get a little bit fruit. So for aims, it will help plants to become more adapted to the dry condition. If you want the plant to become more resistant to drought, you have to add on the element flow which water enter. Let me give you an example. You are in your bathroom, and you know that you have enough water in your tank. When you open the tap, you don't get enough water. The question is, maybe your tube have a problem. Okay? By adding at that tube, you can solve the supply of water. That's the process. Did that answer your question, or not clear?
Yeah, so you have aquaporins in all the cells.
Yeah, aquaporins in all the cells. Water through that aquaporin to answer the plant. You want plant to become more resistant to drought. You want them to become like this, as I said. Sorry. You want to increase the slope of this graph and have something like that. So you want the plant to use a little water to give more fruits, just add on the way through which water enters the plant. And before acting on that element, that way, that canal, you have to know them. What are their role, what are their functional role, what are their phenotypic role. So my current study is to know their role, their functional role, their phenotyping role, their genetic role in tomato. And then I can propose some screening, some selection, some method to improve the water use efficiency of tomato. And this study can be also the basic other scientists can use to work on other species. So in the world, all scientists know that aquaporin is in the plant, in the cell, but no one think about all that. No one knows their role, the functional role. No one knows that.
Good now. This is Maria from Technium. I have a question from the point of view of farmers. Like, even if you introduce all this modification in the plants and then you try to produce them, from their point, do they need to modify something in the way that they cultivate them? Do they need to change the amount of time that are on the light, water? Of course, water for sure, but fertilizers or something like that?
Thank you for your question. Currently, my aim is not to bring GMO to the farmer. Currently, I use GMO technique, CRISPR technique, to study the role of aquaporin in plants. Okay? When I understand that, it's a functional role. You have to know the role. When I know that, okay, these aquaporins are very useful and important, then I can propose many different strategies, many different ways to get the best plants. I know that GMO is ethical discussion, someone has agreed, someone not, but currently my job is not to give GMO to the farmer, no. Currently I want to understand the role of aquaporin. In the future, maybe me or another scientist can say that, okay, based on Hermann Prodjinoto works, then we can propose this solution. Welcome.
Hermann, our time is up. Thank you very much.
Thank you.